I'm very happy for multiple reasons. One is that this is the first time our department is getting this award. Okay, so I'm very happy about that. And more importantly, that may be a trivial reason, but more importantly, I think that this technology is, in my view, meets an unmet need in our country. I don't see if there are any other products or if there are anybody else who will be interested in developing this product. This is in my perception. Also, I think Debjani is probably technically savvy, but business-wise she may not be very savvy, because I don't think this product will make a lot of money for the person who employs it. Okay, so there is, if suppose somebody licenses it, I don't know how much money they get. So since it is not such a great profitable one, there may not be many people who sort of employ it. So I'm also keen on looking at how this sort of gets commercialized, because nowadays if there is money, people take it. Okay, there is money, people take it. So we have to see that. It's very simple. She is going to talk about her technology, so I will not spend time on that. But very briefly, she did PhD from ISC Bangalore. Okay, so my alma mater also stayed. So the non-ISC people say that there is a ISC mafia here. So it is true that we belong to that mafia. She spent about 4 years in France and UK doing postdoc. Briefly, she was in Piramal, and then she joined us about 10 years ago. And she has been doing extremely well, leading a very active research group. And as can be expected, Institute has given her the additional responsibility of managing the Wadhvani Research Center in bioengineering as professor in charge. I think about 1 year ago she started that. So, Debjani, I'm very happy. And there are actually a lot of need for screening sickle cell anemia. And I hope that we will be able to clear all the regulatory requirements so that the product will be in the field as actually that can be used. Okay, so thank you.
Thank you, Professor Atre, Professor Bhandarkar, Professor Balaji for the introduction. I am extremely honored to receive this award, because in the past I have sat in the audience and listened to the awardee lectures, and so it's really a moment of pride for me to be giving this lecture today. So before I go into my talk, and I will have a formal acknowledgement slide, but I just want to say that although I am giving the talk today, this is an entirely team effort. There's a team of students and project staffs, particularly MTech students and project staffs, who have led to this development of the technology. IIT Bombay is the entire system that encourages, very encouraging colleagues in BSB throughout. I'm a non-biologist. The fact that I could work on this, all of that has been possible because of the place where I'm at today. So, okay. So let me just start with the technology. So, what is sickle cell disease, right? This is a genetic disorder. So, what happens in this disease is that we all have red blood cells in our body, and it carries a molecule called hemoglobin. And it is that hemoglobin that allows us to transfer oxygen from one part of the body to another part. Most of the people, they have normal hemoglobin, okay? And we are normally, hopefully disease free. But then sometimes what happens is this hemoglobin changes, which is called hemoglobinopathy. So it leads to various kinds of diseases. For example, thalassemia. Sickle cell disease is another such disorder which affects our hemoglobin. Okay, so it's inherited. That means if our parents have it, then we are very likely to inherit it. Okay. Now, how we inherit it, it depends on whether both the parents have it or just one of them have it. Okay. If I inherit both what are called sickle genes, which code for this abnormal hemoglobin, then we have what is called the full manifestation of the disease, or sickle cell anemia. On the other hand, if only one parent has a sickle cell gene and the other has a normal hemoglobin gene, then we have partially this disease. So that is called sickle cell trait, and it's very important to understand the distinction between these two, because it decides going forward how that person lives their life. Okay. Now, why is this hemoglobin special? Because when you have sickle hemoglobin and you go under a low oxygen concentration, for example, when your blood flows from artery to the veins where the oxygen is less, right, it polymerizes. And what happens then is that when you have this normal biconcave red blood cells, which are very soft in our body, the moment hemoglobin polymerizes, they take this kind of a sickle shape. That is why it's called sickle cell disease. These cells, they are very stiff. So what they do, they enter the blood vessels, and normally our blood vessels towards the part, capillaries, where they, the gas exchange happens, they are about the same size as that of a red blood cell. They're actually somewhat smaller. And red blood cells being very soft, they can squeeze through these vessels. But the moment you have these sickle cells, they're so stiff and they're very sticky, they kind of stick to each other, they end up jamming these blood vessels. Now, how that manifests in the person, that depends on how we have inherited the disease. If I have sickle cell anemia, then this person would have recurring pain, and this is unbearable pain, joint pain, which resembles arthritis, but it can be like somebody as young as a 20-year-old a person. You have anemia because these blood cells are very brittle, they break, and you end up getting that. Ultimately, most of the organs are damaged and leads to very early death. So people who get sickle cell disease, the rate of child mortality is very high because they're prone to infections and so on. And even beyond 20, people don't really live to be beyond 20, unfortunately. That's how bad it is. So who does it affect? And if you see, this is a map that made by Wellcome Trust. The red parts indicate the areas of the world where sickle cell disease is very common. So you can see most of Africa, especially Western Africa, sickle cell disease is very common. The Arabian Peninsula, even parts of India. Okay, so this is not as uncommon as we may think. It's pretty common. I started this project in 2014, right? So when I was writing my grant proposal, I was doing Google search. And these are the Times of India headlines that I came up, right? So in Maharashtra, especially in the Vidarbha region, sickle cell disease is very, very common. So 2013, the headline says that there is no research center in Vidarbha yet, although this is a very common problem. 2017, another search, same Times of India, and it says another promise for a sickle cell research center in Vidarbha. Now, fortunately, very recently, something has come up, but it will take us a long time for this treatment. So there is no cure for this disease, by the way. So you can do an early diagnosis and you can actually help people manage the disease, but there is no cure at the moment. People are working on stem cell treatments and so on to come up with a cure. So this is really a big problem, as 105 million people are affected in India. Most cases are undiagnosed. The way I started working on this problem is actually very interesting. So there was a group of visitors to our department from the Debjani Foundation, and in the talk they said, why don't you work on something that affects India per se? And they mentioned, why not, for example, sickle cell disease? That's the first time I heard about it. So then I started reading up, and then we fortunately got the grant and we started working on it, right? But a lot of people, this is not really known. So now this is a disease where the blood cells change their shapes. Then obviously if you look under a microscope, you should be able to tell what is a sickle cell, right? Or what is a sickle cell blood sample. But there's a problem. Sickle cell was first discovered in 1940. Okay, so under low oxygen environment, somebody showed that the cells become sickle. So they look like this. And since then, there's been a lot of efforts with microscopy. Even Linus Pauling published a few papers on looking at microscopy tests for sickle cell anemia. The reason that did not really become popular among clinicians is that if you look at this image, and unless I showed you with this red circle, there's a sickle cell over here, it would be very difficult to find out. This is actually the blood from a sickle cell patient, and I'll show you in a bit how we can change that. So this is the reason why microscopy is not used today. So how is it diagnosed? And okay, before I go into that, I just also want to tell you there's a whole bunch of work that's going on with deep learning, various complicated image processing techniques, etc. And you'll see a lot of papers coming out. But the key problem that we are trying to solve today, which is distinguishing between what we call trait and anemia, none of these microscopy tests are able to do. And there's a reason for that, which I'll explain in a bit. Okay, so that is the problem that we are trying to solve. So the way it's diagnosed today is that you first, so this by the way affects people who live in very remote areas. They belong to certain tribes and there are no hospitals nearby, right? So you go to those sites people from the screening camps or government health screening programs, they take a finger prick blood, they mix it with this chemical, and they try to see if the blood becomes cloudy or if it's still clear. If it becomes cloudy, all it tells you, you have some hemoglobin disorder. It doesn't say whether you have sickle cell disease or you have thalassemia or something else. To do that, you need another test which is called high performance liquid chromatography or HPLC. So for that, what they do is that they take 4 ml of venous blood, so they take from a venipuncture, then they transport those blood samples to different centers where they do that, and then this result comes back. Okay? Now, when we were doing our test, we were doing some survey to see where you can do this. For example, even in a city like Nagpur, which is a town, right? It's even if you get these blood tests, the blood samples are still sent to Mumbai, and it takes at least 48 hours for those reports to come back. And they will not run this test for just one sample. It's just not economically viable, right? So they'll wait for a good number of samples to accumulate, and then you will get your results. So all of that delays the treatment. The facilities are not there in endemic region, and there's a real social issue is that somebody may be screened and be found to be having some hemoglobinopathy. But the fact that they have to travel to this hospital to get the second level of test, it doesn't happen. There's a lot of lack of patient compliance, and because you lose your wages and so on, and it's a fact that people just don't come back. They'd rather live with the disease than lose wages. So that led us to think that how can we come up with a microscopy test that can be done at point of care and that can distinguish between trait and disease. So that was the challenge that we were working on. So before I explain the details, let me give you an overview of our technology. Okay? So what we do, we take a drop of blood. So you don't need venous blood here, a finger prick will do. Then what you do, you mix it in 2 tubes, okay, with it's just like this. So you have these 2 tubes here. So you have some chemicals, you open them, you add some drop of blood, you shake this, and then what you do is you add that blood into 2 of these imaging chips. Then you set it aside for 30 minutes, and meanwhile you take another patient sample and you do this. After 30-minute elapses, you take these 2, and this is the mobile microscope that we had to develop to work in the field. You take an image of these 2 slides like this, and then you compare these 2 images and you do some image analysis to say whether it's trait, healthy or disease. Okay, so there are 4 parts to this entire technology, and I'll run you through all of that. The advantage is you can get results within an hour. There is no need for blood storage and transport, and even with this microscope you can actually do it in even collection centers. Okay. And the key part of all of this is the most important thing that allows us to distinguish between trait and disease, because here we actually control the hemoglobin polymerization. Whether it polymerizes slowly or fast, that changes the result and the cell shape. Okay? Okay, so where do we get the blood samples? When I started, I am trained as a physicist. I have no idea of biology, and I had wonderful colleagues who connected me to various hospitals in Mumbai, and turned out that there are very few sickle cell patients in Mumbai. You can't do that. So what happened was, week after week, my students, through those connections, they would travel from Mumbai to Valsad Raktadan Kendra. So taking some 4 a.m., 4:30 a.m. bus, train, whatever, and they would get there, work with the people who were doing screening of the nearby villages, Navsari and all of those areas, and then they would come back at night and they would process those samples. This is how we worked on those technologies. Here are a couple of images from the early tests, and this is how the microscope, by the way, used to look, and this is what we would just take there for our studies. Okay? Okay, so now let's get into the technology. The first thing is, why do we need a microfluidic chip here to begin with? It's all we are doing is imaging. There is no flow, right? So what matters here? The first thing is that if you take a blood smear, right, like you do here, all of you have seen that, then what happens? Biconcave RBCs, they can lie sideways, like for example here. And an image processing program might actually recognize this as a sickle cell, which is not the case, right? You want it to be actually very correct. So biconcave means they have to lie flat, okay? So the way a microfluidic chip helps is that we have a glass-on-glass chip with this yellow thing as a spacer. This is the side view, and we basically played with different heights of the chip, and we found that if we reduce the height of the chip, then multiple things can happen. You can have these blood cells lying flat, as well as you also want to see how fast can you get this sickling reaction to happen, right? And because of this confinement, I think that you see some sort of this TD is the time it takes for the first cell to sickle, and you can see that it sort of depends on the chip height. The lower it is, the quicker the reaction happens. And this is basically, you also don't want to make the chips so tiny that you have only very few cells in the field of view. So you also want to have a decent number of cells in the field of view to get decent statistics. I won't go into too much of these details here. I can take them in questions, but effectively we optimize that if you want to kind of have a trade-off between these two things, a 30 micron chip would work for us. So that's what we did. Then the next question was, when we started, we wanted a microscope that we can take to the field with us and take good images, transfer them and so on. And there was no microscope available. Like all the analog microscopes that we have, they didn't really work for us. And so we had to build our own microscope. So one of my MTech students, Samrat, he's joined 2014 itself, started working on this microscope. And this is the microscope that he built in 2015. This is what it looked like when he graduated. Incidentally, because this microscope was needed for this project, we also independently kept on development, and it went through various and many, some 12, 13 stages of that. And today we actually have 2 market-ready prototypes for it. The small one which you see here is for sickle cell disease, but the bigger one that you see here, it's a regular lab-ready microscope. So we recently licensed these 2 technologies that has just come out of this project, okay? So that's one. Then the question is, how does sickling look? So we built a microscope. What does it look in real time? So let me show you in a video. It's the same blood sample that you saw a figure of. So, okay, so you see this is a speeded up video in 30 minutes. There was that thing that you saw in the beginning. At the end of 30 minutes, because you are in the way you have optimized the chemistry and you are using this microfluidic chip, that after 30 minutes you have so many sickle cells that there is no way for you to mistake that this belongs to a sickle blood. Okay, so this is the image that I showed you in the beginning, and this is what it looks like after 30 minutes. So, okay, in this way you can tell that whether it is a sickle blood or a normal blood, but I still haven't told you how you would figure out whether it's trait or disease. That's the key thing, and that's where we have to understand a little bit about how hemoglobin polymerization happens. So again, this is not something that we came up with. There's been a lot of studies since 1970s on sickle hemoglobin, which they have specifically looked at it as a polymerization process, okay? So they found out that if you take sickle hemoglobin, and depending on whether you slowly polymerize it or you polymerize it very fast, like quenching, then the kind of long fibers that you get, that changes. When slow polymerization leads to a few of these very, very long fibers, okay? And that leads to this sickle shape that we are very familiar with. Some intermediate shape speed leads to this sort of holly leaf shape. There are multiple smaller fibers, and very fast polymerization, these don't really get to grow, so they become like this granular shape, which is very similar to unsickle shape and so on. So what we realized, one thing is that we can actually control the speed of polymerization by adding a chemical in 2 different concentrations. If I have a high concentration, speed of polymerization will be high. If I have a low concentration, the speed of polymerization will be low, okay? So we worked on it. The other thing that we knew was that whether somebody is a trait or a disease, the sickle hemoglobin varies in them. For people who have sickle cell anemia, their almost entirety of the hemoglobin is sickle hemoglobin. There's absolutely no normal hemoglobin. For somebody who has trait, they have partially sickle hemoglobin and partially normal hemoglobin. So the amount of sickle hemoglobin matters. And I'm not going into these 2 things. Now, these are the 2 things that we knew. So what we found can be summarized in this cartoon here. So you take a red blood cell. Now, if you have somebody who has sickle cell anemia, which means your HbS stands for sickle hemoglobin, and green up triangle means it's high. If you have that, when you have fast oxygen removal, you get granular shapes. When you remove oxygen slowly, you get these kinds of shape, okay? On the other hand, for trait, somebody who has only partial sickle hemoglobin and partial normal hemoglobin, fast and slow oxygen removal again gives some very different kinds of shape. Okay, so this is what is called a holy leaf shape, and this is almost looks like unsickled when you have very slow oxygen removal. So that tells us that every time, if its sample is sickle trait or sickle disease, I get very different kinds of cell shapes, provided I remove oxygen slowly or fast. Okay, so that's what we have. By the way, these two are two more MTech students who kept on working on this and came up with this technology. Now, just by looking at eye, it might not be very easy to tell, and this is where we need to do some image processing. So what do we do? So we get two images that look like this. So let's say I have a trait sample which looks with high concentration of this chemical, which means fast oxygen removal, I get cell shapes that look like this. And when I have very low oxygen removal, very slow oxygen removal, it looks like this. Okay. What I do then, for each of these red blood cells, I can calculate this quantity called roundness. It's basically a measure of how circular this is. And the reason we use roundness is we are using bright field images, no staining, and we don't need to know the measure the perimeter of the image that well. You can only get the major axis and it works. So that's why roundness is a better measure than, say, circularity. And these are the different kind of roundness values that you can have associated with these cells. Then what we do for an image like this, and it's just a cropped up part, usually you have about 100-150 such cells, you plot the distribution as a function of the roundness. So roundness goes from 0 to 1 and you plot these frequencies, okay? And these curve shapes are very different in each of these cases. So that's what we use. So what do we do then? Take any of these two curves, right? Let's say they look like this. You take a portion of that curve, okay? So let's say the red part and the blue part, and you measure the area under this curve, okay? And this, let's call these two parameters P1 and P2. So every image, first of all, they give you a curve like this, and then you reduce each curve like this to a point with coordinates P1 and P2, right? Therefore, if you now plot them, this P2 versus P1 in a plot, these points would lie on different places. Depending on what the most of the cell shapes are, they would lie on different places in this. Now over here, we had about some 200 odd known samples. Known means they were told that this is disease, trait or healthy samples, and we plotted them over here. And you can see that the blues and the reds are the sickle samples, they group together. The blacks are the healthy samples and they group together. And then you do some sort of clustering and you can get this dotted line, the equation of that, okay? Now this sort of gives you a phase space like picture, okay? So what tells you is that sickle blood samples, they group on this side, healthy blood samples, they group on this side. What do we see further? We see here is that now remember, we have two images, right? So for every blood sample, we have such 2 points. So if both the points, they lie in this gray region, then this is a healthy sample. If both the points lie in this white region, we find that this is a disease sample. If one lies here and the other lies here, then it's a trait sample. You can see it better. So both for high concentration and low concentration, the red ones lie here, the black ones lie entirely here, and for the blue ones, one lies here, one the other. Of course, you may ask what happens for blue if this order switches. Well, it may happen, very rare, but we just have to call it an invalid test. We can't use that, okay? So this is the classifier that we developed. So over the years, we worked with many samples, and this is what we finally came up with. And then we wanted to validate this classifier with some unknown samples. This was a pilot study, very few samples, but we see that when you have healthy samples, they actually fall here as we expect. When you have disease samples, they fall here, and when you have trait samples, they fall very closely here, okay? So this is how you can actually have a validation of a result. Now please note, all of these were done in my lab. So we would go to Valsad, bring back the blood samples, and we would test them here. So the next step of the work was for us to see what happens if we go to the field when a screening camp is going on. Does this work? Because there may be many changes here. The day temperature varies during the day. We went to Dahanu where the difference between day and the night temperature in December was quite a lot. And polymerization is supposed to be sensitive to temperature. Does that matter? You don't have a closed lab, a controlled environment, all of those things. Does it affect anything? So we went to Dahanu, we went to a school, we went to 9 schools for 3 weeks. So students, they stayed there. We sent them a car full of provisions and samples. They stayed in a hospital in the middle of nowhere for 3 weeks, and they kind of did these tests. Similarly, we did some tests in maternity homes in Nagpur, and I'm going to show you a clip from these screening camps to get an idea of what we did. What I have given you, these results are with a very small number of samples. If I have given you the impression that the technology is today ready for a doctor to be taken up, that's not the case. So what I'm showing here are the chips that we make in the lab today, and this is not how one goes for a large scale trial. So the microscope is ready, but rest of the things we need to have prototypes developed, okay? So we need to have a way to make microfluidic chip in the way we want commercially. We need to figure out a way how we store these chemicals with the shelf life, over months, so that people can do these tests and so on. The image processing today I'm doing on a computer, I want to do it on a mobile phone, and all of this finally has to be in a form that somebody with minimal training can do this. So if we can do this, then hopefully we can replace HPLC and we can give confirmed results. So that benchmarking also has to be done with a large number of samples in different parts of India. So that's very, very important. And once these things are there, as Professor Balaji mentioned, we will think about how we transfer this technology, okay? So I have covered only in a very shallow way, just the overview of the technology, but the more details are given in this preprint here, and you can sort of go into more details if you can see. And finally, I started by acknowledging IIT Bombay, but let me now specifically acknowledge everybody. So Claudie, Riddho, Samrat, and so these, who am I missing? And Pratiksha, yes. These 4 were MTech students. These were parts of their MTech projects over the years that they have worked on it. Most of these people, this entire batch except Santosh, they have all graduated and moved away, but I'm very happy today we have Oshin here. She also went away, did her PhD, just coincidentally she's here today, so she's sitting there. Minakshi, Srishti, Anish, Sampath, Sahar, Priyanka, Santosh. It's a huge teamwork that has made this possible. The project initially was started by Bill and Melinda Gates Foundation. They gave a Grand Challenge Explorations Award. Then the Tata Center took it up primarily for development of the mobile phone microscope, but they also funded the sickle cell, some of the field trials. And currently we are supported by WRCB through the Wadhwani Foundation. And of course we have had IIT Bombay's seed grant has been extremely generous and flexible. I made it last as long as I could to support phases when there was no external grant. For blood samples, I'd like to thank Valsad Raktadan Kendra, Talasari, Dayanand Hospital, and finally Sickle Cell Association of India, and all the patients who have actually agreed, signed consent forms and given us their blood. And thank you very much.